Hello students, let us learn about incorporation of recombinant DNA into cells. In introduction, recombinant DNA that is generated by means of genetic engineering need to be introduced into host cells for the expression or multiplication of the gene of interest. There are various methods of introducing a recombinant DNA into host cells, the selection of which is dependent on the type of the host that is to be used. As for instance, calcium chloride method is an efficient method in transformation of prokaryotes but does not function in plants and other eukaryotic cells. Similarly, microinjection is not possible in E. coli cells due to its size. Therefore, understanding the technique of gene transfer and its application will enable us to make better choice of transforming techniques. At first, let us discuss about the calcium chloride method of transformation. The ability to transform bacteria can be traced back to 1928 with Frederick Griffith's experiment using Streptococcus pneumoniae, a bacterium that causes respiratory disease in mammals. Griffith called the phenomenon transformation. Today, chemically induced transformation is commonly used in many laboratories. Some strains of bacteria are not naturally transformable but can be artificially induced to take up DNA by chemical or electrical shock methods. In the early 1970s, Mendel and Higa demonstrated that bacteriophage DNA can be transferred to E. coli K12 strain, a strain that is not naturally transformable by addition of calcium chloride. 3 years later, Cohen and Chang showed that circular, nick-circular or shared DNA can also be assimilated by bacterial cells and recovered in a covalently circular form. Since then, many laboratories have modified the original method with the goal of increasing transformation efficiency. Chemically induced competence followed by transformation is a commonly used technique to introduce plasmids or other DNA fragment into E. coli. Depending on the genetic information it carries, the incoming DNA can be replicated as an independent entity or integrated into the host chromosome. To select for cells that incorporate the DNA, a plasmid is engineered to carry a selectable marker such as antibiotic resistance genes. Plasmid uptake provides the host cell with the ability to survive on a selective media. Today, chemically induced transformation is frequently used to clone and amplify a fragment of a gene, a whole gene, or an entire DNA library. Transformation of cells with DNA is an invaluable technique that provides scientists with a way to introduce and manipulate genes. Since E. coli is not naturally transformable, the ability to take up DNA or competency must be induced by chemical methods using divalent and multivalent cations of calcium, magnesium, manganese, rubidium, or hexamine cobalt. Alteration in the permeability of the membrane allows the DNA to cross the cell envelope of E. coli, which is encomposed of the outer membrane and inner membrane and a cell wall. The outer membrane of E. coli can be understood by application of the fluid mosaic model for membrane and is composed of phospholipids, proteins, and lipopolysaccharides. Many channels or zones of adhesions are formed by the fusion of the outer membrane and the inner membrane through the cell wall layer. Although the transformation mechanism is not known, previous studies indicate that these channels allow for the transport of DNA molecule across the cell membrane. The negative charge of the incoming DNA, however, are repelled by the negatively charged portion of the macromolecules on the bacterium's outer surface. The addition of calcium chloride serves to neutralize the unfavorable interactions between the DNA and the polyanions of the outer layer. The DNA and competent cells are further incubated on ice for 30 minutes to stabilize the lipid membrane and allow for increased interactions between calcium ion and the negative component of the cell. The reaction mixture is then exposed to a brief period of heat shock at 42 degrees centigrade. The change in temperature alters the fluidity of the semi-crystalline membrane state achieved at 0 degrees centigrade, thus allowing the DNA molecule to enter the cell through the zone of adhesion. Other factors also affect transformation efficiency. Prior studies show that some E. coli strains are more susceptible to transformation than others due to difference in the composition of the lipopolysaccharide. E. coli with a lengthy O-linked polysaccharide blocks or hinders DNA from entering the cell. Adding magnesium to the media increases transformation yield by enhancing the ionic interaction of the molecules on the surface and therefore alters the suppleness of the membrane for more efficient transformation. In the next method of transferring recombinant DNA, we have electroporation. Electroporation is the use of high voltage electric shocks to introduce DNA into cells. It is a procedure that has gained in popularity for standard gene transfer and also allows the generation of genetically modified mice. It can be used with most cell types, yields a high frequency of both stable transformation and transient gene expression, and because it requires fewer steps, it can be easier than alternate techniques. Electroporation can be used for both transient and stable transfection of mammalian cells. Cells are placed in suspension in an appropriate electroporation buffer and put in an electroporation cuvette. DNA is added, the cuvette is connected to a power supply, and the cells are subjected to a high voltage electrical pulse of defined magnitude and length. The cells are then allowed to recover briefly before they are placed in a normal non-selecting cell growth medium. Selection for permanently transfected cells and for cells carrying targeted genes insertions by homologous recombination can be accomplished by modified media. The next method of gene transfer is the particle bombardment or biolistic method. Some cells, tissues, and intracellular organelles are impermeable to foreign DNA, especially plant cells. Biolistics, including particle bombardment, is a commonly used method for genetic transformation of plants and other organisms. To resolve this problem in gene transfer, the gene gun was made by Klein at Cornell University in 1987. On the gene gun technique, Klein and Stanford et al. Published papers, obtained patents, and formed a company called Biolistics. The gene gun is part of the gene transfer method called the biolistic method. In this method, DNA or RNA adhere to a biological inert particles. By this method, DNA particle complex is put on the top location of target tissues in a vacuum condition and accelerated by powerful shot to the tissues. Then, DNA will be effectively introduced into the target cells. Uncoated metal particles could also be shot through a solution containing DNA surrounding the cell, thus picking up the genetic material and proceeding into the living cell. The efficiency of the gene gun transfer could be dependent on the following factors such as cell type, cell growth condition, culture medium, gene gun ammunition type, gene gun settings, and experimental experiences, etc. In virus-mediated gene transfer, this method involves the introduction of genes into host cell's genome using a virus as carrier. The viruses are used in gene transfer due to the following features. First, efficiency of virus to deliver the nucleic acid into cells. Secondly, high level of replication and gene expression. The foreign gene is packaged into the virus particle to enter the host cell. The entry of virus particle containing the candidate gene sequences into the cell and then to the nuclear genome is a receptor-mediated process. The vector genome undergoes complex processing, ending up with a double-stranded DNA depending on the vector that can persist as an episome or integrate into the host genome, followed by the expression of the candidate gene. In another way of transferring recombinant DNA, we have lipofection. Lipofection or liposome transfection is a technique used to inject genetic material into a cell by means of liposomes, which are vesicles that can easily merge with the cell membrane since they are both made of phospholipid bilayers. Lipofection generally uses a positive charged cationic lipid to form an aggregate with the negatively charged or anionic genetic material. A net positive charge on this aggregate has been assumed to increase the effectiveness of transfection through the negatively charged phospholipid bilayer. In the last method, we have microinjection. Microinjection is a technique of delivering foreign DNA into a living cell, a cell or egg or oocyte or embryos of animals, through a glass micropipette. One end of a glass micropipette is heated until the glass becomes somewhat liquefied. It is quickly stretched, which forms a very fine tip at the heated end. The tip of the pipette attains to about 0.5 micrometer diameter, which resembles an injection needle. The process of delivering foreign DNA is done under a powerful microscope. Cells to be microinjected are placed in a container. A holding pipette is placed in the field of view of the microscope. The holding pipette holds a target cells at the tip when gently sucked. The tip of the micropipette is injected through the membrane of the cell. Content of the needle are delivered into the cytoplasm and the empty needle is taken out. One of the successful experiments with microinjection is the transformation of oocytes of Xenopus. Xenopus oocytes have been widely used for the study of transcription by microinjection because oocytes contain between 6,000 and 100,000 or more RNA polymerase molecules than somatic cells. Microinjection is technically easy because of large size of oocyte. Some of the endogenous pattern of gene regulation during development has been characterized. The injected DNA integrates randomly into nuclear DNA, and its expression could be possible only when the foreign DNA is attached to a suitable promoter sequence. There are many examples where different types of animal cells have been microinjected and successfully transferred. In conclusion, we have learned about various DNA incorporation techniques in living systems. Careful and appropriate selection of transfer technique is essential for successful cloning and expression experiments. However, the technique of recombinant DNA technology doesn't end with the incorporation of DNA into host cells. There is yet a process of selection of transformed cells from non-transformants. After selection of transformants, the host cells are ready to be processed for other applications of biotechnology.